the webinar. So good morning, good afternoon, and good evening, everyone, and welcome to today's Bioturing webinar. So we are honored to host James Pearce, a scientist at Chan Zuckerberg Initiative, who is leading a groundbreaking work at the intersection of machine learning and biology, which is, James is the first author of TranscriptFormer. So before we talk about TranscriptFormer, um, James Pearce has a PhD in particle physics, where he developed machine learning models to analyze data from the Large Hadron Collider, right? Which is, I think, the world's largest and highest energy particle accelerator that lies, beneath the France and Switzerland border near Geneva,
Mm.
if I'm not making mistakes.
Yeah, that's right.
So since then, he worked as a applied scientist in tech industry, in many different companies, building models for search and natural language understanding. So if now you call your Alexa and you have problem with that, you know who to blame. And Jem is now at the Chan Zuckerberg Initiative, building virtual cell models, to better understand biology with AI. So Jem is the first author of the TranscriptFormer. This is the main topic of the discussion today, which is a cross-species generative model trained on over, I think, 110 million single cells from 12 species. These 12 species spanning more than 1.5 billion years of evolutions. I think it is quite a bold attempt to build a foundation model of single cell transcriptomic, like embedding genes and cell in a shared space, prompting biological knowledge and transferring insights across species, and also assays and perturbations as well. So without further delay, please join me welcoming James as he will take us to the future of generative biology. Welcome, James.
Awesome. Thank you, Son. Thanks for having me. I'm really happy to be here and excited to share the work that we've been doing on TranscriptFormer here at CZI.
Mm.
So, if you guys can see my screen okay, I'll just jump into it.
Yeah.
So before I start talking about the paper and the model, I'm going to first introduce the team. So we are a multidisciplinary team of scientists here at CZI, and we are AI scientists, we are biologists, engineers, data scientists, and we have a TPM here as well. And I think when we work on a big project like this, really is important to have this diverse team in terms of backgrounds. And so while I'll be giving the presentation today, it really is a team effort, and credit goes to the whole team. So thank you. So yeah, let's get into it. So, like you said, TranscriptFormer, it's actually a family because we train three different models under this name. But it's a family of single cell foundation models trained on single cell RNA-seq data. So it's not the first single cell foundation model out there. It's similar in vein to something like SCGPT, but what's unique about it is it's trained across multiple species. And so you can use this model on really any species you want. We train on 12 different species, a diverse set of species spanning 1.5 billion years of evolution, everything from human to fungus, etc. And it's also a generative model. So it's a generative model over genes and their expression values. And we use a transformer-based architecture where we tokenize the genes with ESM2 embeddings. So this is similar to what UCE did, which I'll discuss later. And the output of this model, there's actually two outputs. So we have embeddings, which come in two different flavors. We have cell embeddings, but we also get these gene embeddings as well. And then you can also use the more generative properties of the model. So you can get distributions over genes and counts as well. And so I'm going to talk about that a little bit more at the end of the presentation because I think that's a more unique, interesting attribute of this model. So we trained three different models. We have TF metazoa, which was trained on 12 different species across 112 million unique cells. Then we have TF exemplar. This is human and four model organisms, and then TF sapiens, which is just human only. And so when we train these models, we try to keep them as similar to one another as possible, only changing the diversity of the data in the species it's trained on. So each model is trained on roughly the same number of total tokens. So number of epochs may change, but we try to keep the total tokens it's seen during training the same. Although if it's trained on, say, one species, it's going to see more repeated tokens than on multiple species. We also try to keep the number of parameters the same as best we can. So all the parameters in the transformer layers are exactly the same. It's about 300 million parameters in the transformer layers. And there's going to be differences in the decoder head, but that just has to do with the vocabulary. So that changes the decoder head size. And so if you look to the right here on this table, you can see that um, the data is actually very unbalanced. So we have a lot of skew towards human and mouse with not a huge amount of cells from other species. We wish we had more from these species, but that's just kind of what is available right now. So to try to kind of correct this imbalance, what we do is we use this sampling technique it's actually borrowed from multilingual language models. So when you train on multiple languages, they tend to upsample low resource languages. So what that means is when they sample a batch, they're more likely to sample from a low resource language or low, in this case, low resource species. And you can kind of tune how likely we're going to be sampling from that species with this hyperparameter alpha. And the reason you want to tune that is because you don't want to overfit to one of these species by sampling from it too much. And so during training, you want to tune your mix for each species. So each one is represented as best it can be without overfitting to any one of these species. This is the training technique we use there.
Right.
So now I'm just going to say briefly about the data. This is, like I said, single cell RNA-seq data. Probably most people are familiar with it, but the way I like to think about this data is it's kind of modeling a cell as a bag of transcripts, right? So what we get out of this data is we know how many times we've read each type of transcript. A transcript can be associated with a gene. And so we can also represent this data as basically a histogram. So we look at each gene, we have some count value associated with it, and that represents the cell. So I think this is the most natural way to model the data as basically a histogram. And you can kind of think of this generative process as we first model the genes as a categorical distribution. So then we can sample a gene from this categorical distribution. And then once we know the gene, then we can sample the counts from a Poisson distribution. And so that's basically what this is showing here. We use a zero truncated Poisson distribution because we don't include the zero count genes. So if the gene's not expressed, it's not part of our input to the model. This is just kind of more of an efficiency thing.
Mm.
I'll discuss more on that in a bit. And as I mentioned earlier, we're using ESM2 to tokenize the input genes. And so the reason we do that is because we want to define this vocabulary over all species. So the typical way you do tokenization is you would say tokenize every gene for every species. And that actually works fine as long as the species is within your vocabulary, which is to say in the training set. Now we trained on 12 species, but we wanted to test on much more than that. So we need some sort of generalized way to create gene tokens. And so some sort of protein language model is kind of a good fit here. And so the way this works is for a given gene, we first find its associated proteins or proteins in isoforms. And of course, that can be encoded into ESM through their amino acid sequence. And so we can get multiple outputs from ESM for a given gene, but then we just average these together to get a single gene level embedding for the input to the model. And I should also say that we have a benchmark model that we call ESM2 cell embedding. And this is our own benchmark that we designed. And basically, we just take the sequence of genes that are encoded by ESM, we then average them together, and we use this as the final embedding for the benchmark model. And the reason we want to have this benchmark is we want to be able to measure how well TranscriptFormer is doing compared to just the inputs alone, right? Because this basically represents the inputs to the model. Because, of course, ESM2 is already a very powerful model in its own right. So, like I said, it's a generative model, and it's an autoregressive generative model. So it's kind of in the style of a GPT model, where you predict the next token, you're predicting the next gene, and then the next count value. We train with causal masking, which is, I think, fairly unique. A lot of other models don't actually use causal masking. And we also use teacher forcing, which is the typical way of training this. Except we leave out the positional encodings. And so we leave them out because there's kind of no innate sequence here in terms of the genes in our data. And so we don't have them there, but there is a ordering in terms of the sequence in which we feed it into the model. But this order is random. It can be random. We actually randomize it during training. It's kind of like a form of data augmentation. But the model does depend on, so at each position, it does depend on everything in the context. It just doesn't know the order in which the genes are in the context, right? So the order doesn't matter for predicting the next gene, but the context, the set of genes, does matter. And so we also use this assay tokens. We append the context with a token that just tells the model what sequencing technology was used, so 10x versus SmartSeq, etc. But we don't use any other information than that. So no other metadata like cell type, etc. This is just genes and the expression values. And the max sequence length is about 2,000, which, since we only include expressed genes, this is generally enough. But if it's longer than that, we just truncate it. And then there's also here the likelihood we use, which again is just the joint probability between the genes and the count values, and it breaks down via the chain rule here into the categorical and Poisson distribution that I mentioned earlier. So in terms of the architecture, it is a transformer-based model, but there are some unique aspects to it. So first is that we use these two decoder heads. So the typical decoder head is like the gene decoder head, which predicts the next token. But we also have this count decoder head. And the count decoder head is conditioned on or dependent on the next gene. So it doesn't really make sense to predict a count unless you know what gene you're predicting that count for. So it needs to condition on the next generated gene. The way we do this in the model during training is we concatenate basically the input to the transformer layer with the output, where the input is shifted right in terms of the sequence index. It's shifted right one. And so it skips ahead one gene. And then we feed that into the decoder head, and then it can condition on it. And the reason we do have this skip connection here rather than using, again, the output of the transformer layer is because we don't want to leak any information from the next gene into the gene decoder head, right? And so that would happen if we did use the output of the transformer layer. So that's why we use a skip connection. We use pre-normalization, which is pretty standard now. No feed-forward or attention biases are used here. We use an embedding size of 2,000, 16 attention heads, 12 transformer layers, and the total number of parameters, like I said, for the transformer layer is about 300 million for each model. But then depending on the vocabulary size of the training set, the head can be up to an additional 300 million. So when you have large vocabularies, that gene or the decoder head actually becomes very, very large. So one unique thing we do here is how we encode the counts into the model. So there are different ways to do this. Other models like SCGPT, they tend to first project that scalar count value to a vector, which gets added to the embeddings. We took a different approach. So instead, we add the counts into the attention layers as an attention bias term. Actually, the log of the expression values. And so what this does is it basically upweights, or the model pays more attention to the genes that are more highly expressed. Um, and the motivation behind this is actually has to do with the interpretation of the data as a bag of transcripts, where a transcript or a gene is repeated multiple times in the cell. And so if you were to naively just to make a very long input with a context that had repeated versions of each gene token, C times, uh, and fed that into your model, that would be one way of encoding the count information. But here, this is basically a more efficient way of doing that. So you can show that if these tokens are in fact identical, and they in this case they are because we're not using positional encodings. This is equivalent to just adding this bias term into the attention layer. And so it's just a more efficient way of representing those repeated tokens. I
M,
should say yeah, you have a question?
M, M is what? Big M?
Oh, big M, sorry, big M or,
What is M here? C in
oh, I see. See, yeah, yeah, sorry about that. M would be, I guess, here the sequence length, right? So it's the same dimension as the the arguments to the softmax, or
okay.
the attention matrix. So yeah, so TranscriptFormer, it has two different embeddings that it outputs here. So you can get gene embeddings or you can get cell embeddings. And the gene embeddings, so basically the input to TranscriptFormer, has some sort of sequence. So the output has the same sequence, so we can associate every input gene with an output. But when we input the genes, they're largely uncorrelated, right? They come from ESM2. In the output of the model, these genes get correlated with one another through the attention mechanisms of the transformer layers. Um, and they're really correlated with all the genes that came before them, right? Because they're conditioned on those. So we call these contextualized gene embeddings. So they're not just embedding the individual genes, but the gene plus the context kind of around it. Um, so typically you wouldn't use them all this way because the output tends to be very large. So if you have 1,000 cells in your data set and you have 1,000 expressed genes per cell, your output is 1 million CGEs, right? Because you get one per cell per gene. Um, but here on the UMAP here, I show one data set where I filtered just to the top 10 genes here, and I show what they look like. And you can see they do cluster by cell type, but they also cluster by gene because they're colored by gene here. So there's some clustering within cell type by gene, which is kind of interesting. So it does still retain some of that information from the original gene, as well as the broader context, which is cool to see. Um, and actually, we see that some of these genes cluster more than others, which is kind of interesting. We don't completely understand that yet, but maybe this is future work to better understand that. So a natural question to ask is, what context do the gene embeddings actually learn, right? And so as I mentioned, cell type is one, but there are other things it could learn as well. And so we did this analysis where we did PCA variance partitioning analysis, where we look at multiple cells and then we group the CGEs by their gene and average over them. So we get average CGEs. And so this kind of helps reduce some of the noise we might get from a single CGE. And then we look at the PCA components, and we find that indeed, most of the variance is actually explained by cell type. So about 95% of the first two principal components are explained by cell type. Some of it, about 2% by tissue, and then some also by donor. But it is pretty interesting to see that these concepts like cell type, tissue, and donor do emerge from the model, even though it's not explicitly trained to recognize cell type or tissue, etc. It's just trained on
So
genes.
cell type was not part of the inputs, right?
Yeah, that's right. Cell type is not part of the input. None of the metadata, except for assay type, is the only metadata we use as part of the input. So
And
um,
why you need to input the assay type?
So mostly because we find different assays have like very different read depth or number of genes, or sorry, number of counts. And so that can really change the count distribution. So it's good to give that information to the model. Um, yeah, and so we can also investigate different levels of aggregation of these embeddings, right? So we can aggregate up to the cell embedding, which I'm going to talk about kind of for the rest of the presentation, but there are other ways to aggregate these embeddings, right? You can aggregate up to the cell type level. So that's what we did here. This is a spermatogenesis data set. So it's a bit of a developmental data set. And you can see that when we aggregate to the cell type level and calculate cosine similarity between these cell type centroids, we kind of reproduce this developmental trajectory in spermatogenesis, starting at male germline stem cell, which is nice to see. And then when we do the same thing across species, so we aggregate to the species level, and this is actually a really nice data set because it has multiple species here. We have a few different primates and then platypus, possum, and chicken. And so all the cell types are also aligned with these, which is great. That's usually not the case. And so we aggregate to the species level and we look at the distance between species centroids. And what we find is that when we plot this similarity score versus the evolutionary distance between those two species, we get um, this nice trend where we show that cosine similarity decreases with evolutionary distance between the species. So, the model we see reveals some of these evolutionary relationships in the data. And I should say this is really interesting because if you simply take the ESM2 embeddings and do the same analysis, you see zero trends. There's no trend here at all, just from ESM2. So this shows the utility and benefit of training on single cell data and using TranscriptFormer here.
And you don't input and species is not also part of the input.
Yeah, exactly. That's right. Species label is not part of the data. We're literally just training on the genes as embedded with ESM2 and the expression values. So it's really great to see properties like evolution kind of emerge from the model.
Nor do you need the homology informations, right?
Yeah, no homology information, nothing like that. Um, yeah, and this was very much on purpose. So first of all, it's a lot easier to collect the data from multiple data sources if you're not trying to align metadata like that. But also we want to see what the model could learn without explicitly training on that information because we would like this model to be useful for actually discovering these types of relationships. Um, yeah, especially new species that maybe are not so well explored. So now I'm going to talk about species transfer learning. Um, so we use this methodology here called reference mapping. And the idea here is that we take a source species, for example, mouse here, which maybe we have labels for. So for example, cell type is a label, say we're using here. And we want to transfer that label information to some target species, which maybe we don't have labels for. Um, so we use this reference mapping technique where basically you can take a query point from the target species and you can find its nearest neighbors in the space of the source embedding species, right? So let's say we find the top five nearest neighbors to that query point. We take majority vote for its label type, and then we map that back to the target species. And then we can do that for every single point in the target species to map the labels from source to target. And so in practice, what this actually looks like is you're training something like a KNN model on the source species and then evaluating on the target species. But this gives us basically the closest thing to zero-shot transfer between one species and another, right? So we apply this method on, again, this is the same spermatogenesis data set, which is a nice testing ground because we have multiple species and we have this cell types that are aligned between them. And we find that TranscriptFormer, both multi-species variants, TF exemplar and TF metazoa, perform very well in this task. And we compare their scores to UCE as well as the ESM2 baseline. So as I mentioned before, UCE, universal cell embedding, this is also a multi-species model trained on ESM2. And so it's in some ways it's a very similar model that we can compare to. And then the ESM2 baseline is mostly just to see how much better we're doing than the information that gets put into TranscriptFormer alone. And so both TranscriptFormer and UC actually vastly outperform just ESM2 alone, which is really nice to see. I think it shows the benefits of training on single cell data. And then if you look at the UMaps here, these are UMaps from TranscriptFormer, I think TF metazoa. You can see that the different primates actually overlap a lot, which I've circled in here in red. So chimpanzee, gorilla, rhesus monkey, and marmoset overlap a lot, which shows that the model's kind of automatically doing some sort of kind of integration between these species. So we didn't perform any explicit integration here. And the cell types they do cluster more or less by cell type, which is nice to see. So these cell types are getting integrated together. We can do this not just for cell type, we can actually transfer any type of label from one species to another. So we did this also on a drug perturbation data set. And these are actually new results that aren't in the preprint yet. So we're going to update the preprint with these new results, but for us to see it here. And so we look at these 4 different species: rat, rabbit, pig, and mouse. Sorry, the cartoon's I guess a little bit wrong here, but we take those 4 species and we have treatment and control. So before they've been given this drug LSB and after, and you can see or so I should say, the task is to try to predict how well we can predict whether or not the treatment was given versus not given, right? But we're trying to transfer these results from one species to the next in the same way. And if you look at the UMaps in the bottom left, you can see the TranscriptFormer models, there's very good separation between the control, which is the purple, and the treatment, which is the blue, green, and yellow. Whereas UCE and ESM2 separation's not quite as good. And so in general, we see very good results here with TranscriptFormer outperforming UCE as well as ESM2 by a large margin, which shows that it does a good job of this species transfer task.
Right.
So yeah, now I'm going to talk about some of the other benchmarks we did, mostly on human. And we used a slightly different protocol for this. So this is often called linear probing. So the idea is we train a logistic regression model on top of the outputs of the embeddings. And we do this for each one of the benchmark models as well. And I think this is kind of a more realistic scenario. This is probably how people are likely to use the model in practice. This is still pretty zero shot, I would say, because we're not fine-tuning any of the models. We're just training this additional simple model on top of TranscriptFormer or the benchmarks. We do this with 5-fold cross-validation. So we do this 5 times and then combine the results for the final estimate as well as the uncertainties. We use some regularization and we don't apply any transformations to the outputs of the models. So when we get the embeddings, we just take those as they are, which I think is the most fair way to compare them. Um, okay, before we go to human, I want to show this generalization to unseen species. So these are species that are largely not in the training distribution. And in fact, a lot of the previous results were also out of distribution species. But here we're looking at full cell atlases from these species that were explicitly held out from the training set. Um, so we did train on human and we did train on zebrafish, but the other ones we haven't trained on. And some of these are very divergent or distant from humans. So stony coral is over 600 million years since the latest common ancestor. Um, and so again, we compare to UCE and the ESM2 baseline. And here what we're doing is we're trying to do cell type classification, and we use this F1 macro score. So we're doing cell type classification for each one of these species individually and looking at their performance versus evolutionary distance from human. And I think we see for all models that we do see that the performance drops the further you go from human, which is probably to be expected because the data is very much skewed towards human, even though these are all technically multi-species models. But TF metazoa and TF exemplar, they tend to remain pretty good and decrease the least as we go out in evolutionary distance. And interestingly, TF metazoa performs a little bit better than TF exemplar here, especially out for stony coral and the more distant species, which maybe suggests that the more diverse data set that was trained on helps here with out of distribution species or generalizing to new ones. Okay, so now I'm going to talk about some of the human benchmarks that we did. So we just um, So for here, we benchmarked against six different benchmark models. So we have the three TranscriptFormer models, but then we also have UCE, SCGPT, GeneFormer, SCVI, which is our own version of SCVI, by the way. It's one that was trained on cell by gene, and so we use this as kind of like a foundation model. Then we have the ESM2 baseline and ADO.cell, which is a newer foundation model. And we use the Tabula Sapiens 2 cell atlas as our held out or evaluation set. We explicitly hold this out of training for TranscriptFormer, but for the other models, it's kind of naturally held out because it's a relatively new um, cell atlas that was just released, I think, this year. So it's not likely in any of the pre-training. So that makes it a good evaluation set. We can use macro F1, which is pretty standard now for these evaluations. We do five-fold cross-validation, but the errors turn out to be very small, so I'm not quoting them here.
Right. So
Um,
the Tabula Sapien 2.0 is the one that you don't put in the training set, right?
yeah, exactly. We make sure it's not in the training set. Tabula Sapiens 1 is in the training set, and there actually is some overlap in terms of the donors, but we were careful to remove those old donors from this evaluation set. So this is actually Tabula Sapiens 2, but it's only the donors that were not in Tabula Sapiens 1. So this is all new donors and should be data that none of these models were actually trained on.
Yes.
So um, we do the linear probing protocol for each tissue individually, and then combine these results. And yeah, I mean, we can see TranscriptFormer does perform best. So TF exemplar actually performs the best with 0.91, and then TF metazoa, and then TF sapiens and UCE actually tie for third, so they both get 0.906. And then we have SCGPT, etc. But I think you'll notice that the results are very kind of saturated on this benchmark. They're all actually very high scores. You can see if you look at the 95th percentile, there is some divergence, I guess, But this is largely dictated by a few points in the tail, which are the more difficult cell types to classify, right? So we
And
looked at some other benchmarks as well. So this one is SARS-CoV, and the task here is to predict whether or not a cell is infected with COVID or not. And we did this same linear probing protocol with 4 different donors and then combined the results. And TF or TranscriptFormer performs very well compared to the other models here. And so all three of those, the TranscriptFormer models show a decent gap in performance compared to UC and SCGPT.
this is for
TF,
what cell type?
this is for I think it is for I think multiple cell types.
Mhm.
I'd have to look up exactly which ones, but yeah.
Yeah.
Um, and so we see pretty good performance across all 4 donors. And if you look at the UMAPs here, you can kind of see why. So there is some separation, I think, between infected and non-infected cells. So these are for TF sapiens.
Mm.
And then so the last benchmark results. So this is again a new result. So we decided to also look at the Tahoe 100 million data set. So this is a relatively new perturbation data set that does drug or small molecule perturbations with 100 million cells. We didn't use all of them, of course. We sampled down to 100,000 cells from plate 3 in the data set. But that leaves us with 95 drug perturbations over 50 cell lines. And then we do again this classification problem. Can we identify control versus treatment? And to make the problem a little bit more difficult, we created a train test split where none of the cell lines in the training set are in the test set, so we separate them. Um, and yeah, again, TF Sapiens does very well in this task, outperforming UCE by quite a bit, and same with SCGPT and the other benchmark models. We didn't actually get all the benchmark models here. We're having trouble getting to work on some of them, which is one of the challenging things about running benchmarks like this.
So
Um,
gene former was not finished.
yeah, so yeah, we're hoping to get the some of the other benchmarks on here as well. Um, but typically we see a pretty similar ranking across these benchmarks. And yeah, again, if you look at the UMAPs here, I think you can see that there's pretty good separation for at least these top drug types between control and treatment.
Mm. Yeah.
Okay, so now I want to talk about the more generative aspects of the model. So as I mentioned earlier, the model doesn't just output embeddings, it also can output distributions over genes. And so far we've only been using and looking at those embeddings, but I think it's also interesting, and there may be some novel use cases here where we can look at those distributions over genes that are completely generative. So the way this works is you can prompt the model similar to the way you would prompt ChatGPT, right? Except you're not prompting it with language, you're prompting it with genes. And so in the simplest case, you can prompt with a single gene, and then you can get a distribution over all the next genes, how probable they all are. You can also generate or use more complicated prompts. For example, you could prompt over multiple genes and multiple expression values, and then get a distribution over all the next genes.
In the second case, does the order of these genes affect the next one?
Yeah, that's a good question. So no, here the orders doesn't matter. It's just the set of genes and their associated expression values that matter. Yeah. So this is one example of a use case. So here we prompt the model with a transcription factor, and then with that transcription factor, we then find the distribution over all associated genes, and we use that to try to identify, interactions between transcription factors and other genes. So we use this metric, which is called pointwise mutual information. So we could have just used the joint distribution between the transcription factor and other genes, but this has the benefit of kind of filtering out uninformative co-occurrences between the two. And it's used a lot in information theory. So we calculate this PMI value, which is completely generated by the model. So we generate the conditional probability here as well as the marginal simply by prompting the model. And then we can calculate p-values for these PMI values. And with these p-values, we can identify statistically significant values. And so that's actually what you're looking at here in the bottom. These spikes here are the p-values, or the negative log of the p-value. So the larger the spike, the more statistically significant that interaction is. We've colored in red we've cross-referenced all the statistically significant values with the string database, which has it's a database of interactions between genes. And so everything in red is a known interaction that was in the string database. Everything in black wasn't in the string database, which isn't necessarily a false positive, but it means that it very well may be. I think it is encouraging to see here that the most significant results very much tend to be in the string database. So it's interesting to see that it really is picking up on these interactions. And and I should further say, like, this is all just generated from the model. So there's no inputs to the model. We just pre-train it, and then after it, we can just interrogate the model and get this information out of it, which is, I think, very cool.
This is cool.
So another way of prompting the model is, like I said, you can use more expressive prompts. And so what we wanted to do here was prompt by essentially a cell type. But for reasons I already explained, we didn't include cell type in the model, so we can't just prompt by cell type. We don't have a cell type token to prompt it with. So instead, what we do is we take marker genes for a given cell type. We calculate these marker genes along with their average expression values, and then we prompt the model with the marker genes for a cell type, and we use that as kind of a proxy to prompting for a cell type. Okay. And then we get the distribution over other genes. In this case, we're looking at the distribution over all the transcription factors, all the human transcription factors that are known. And we're comparing our generated data versus the Tabula sapiens 2 real data. So on the right here, we have Tabula sapiens 2. And what you're looking at is a heat map of the expression values. So the vertical columns here correspond to transcription factors, and the rows correspond to cell types that are associated with those transcription factors, right? And so they've been ordered in this specific way. And the structure you see here is when you see these vertical lines, so that's a transcription factor that is basically always on, right? And so that's called a ubiquitous transcription factor. It's on for all the different cell types. And then for the diagonal trace here, these are cell type specific transcription factors. These tend to be transcription factors that are mostly only on for specific cell types, right?
Each row is a cell or is a cell type?
Each row is a cell, yeah, each row is actually a fine grain cell type, but they are grouped by the broader cell type, right? So the colors you see here on the rows, they correspond to groups of cell types that are in the on the right here. So you can see certain structure here, like for example, F, that's boxed out here. That corresponds to male germline cell types, right? And the transcription factors that are basically just on for those cell types. And what's really interesting is that we see the same structure from TranscriptFormer. And so we didn't actually have exactly the same categorization of cell types. So it's not, we couldn't get to match one to one, but, we did our best to match up the cell types and we still see the same general structure of the vertical bands and the horizontal one, and even the more detailed structure for, male germline, etc., that were pointed out in the paper. We find this as well
So
in
James, the
the
color
generated
on
one.
the heat map on the left and the right, the rows are ordered in the same way, right?
Yeah, yeah, we did our best to order all the rows. So the rows are ordered in the same way. What may be different is the sub-ordering within a cell type. We couldn't find that information, but the broad cell types are ordered in the same way. So we still see like the same general, I think, structure, um, and it
And
reproduces it pretty well.
row there on the left side from TranscriptFormer was actually generated given some marker genes that you say, okay, so this is a six marker gene of this cell, this row, then generate the rest, right?
Exactly. Yeah. So we do generate the marker genes from Tabula sapiens too. So we use that data set to generate the marker genes, but then the rest, the transcription factors, that's completely generated from the model. So that doesn't come from the data itself. And what's also interesting is, like I said, we didn't actually train on Tabula sapiens 2. We trained on Tabula sapiens 1, but not Tabula sapiens 2. So, you know, um, this is in a way doing some prediction as well, which I think is it's very, very impressed. I was actually very impressed with these results when I saw them.
The diagonal lines, and I see the pattern marching on the left and the right side, and I wonder what are the percentage of marker genes that you put there? You know, if you put that 100%, then it's not surprising.
That's true. So we did 100 marker genes, um, but it's not predicting the marker gene rate. It's predicting the transcription factors, the distribution over transcription factors. Um, and so yeah, do need to condition it on the right information. You need to give it information that tells it that it's conditioning on this cell type, or else you won't get the structure. And I think, this is a promising direction. We like in future versions, maybe we think about better how we can prompt the model or how we're going to prompt the model to get this information out. It is a little bit of work to get those marker genes. But yeah, you can kind of think of this like the model is like a very compact representation of the data that it's been trained on. And so you can just interrogate the model itself in hopefully complementary ways to interrogating the data and find these different structures out of the model. Um, so I'm kind of excited about these use cases, as maybe you can tell. Okay, so that's my presentation. Thank you all. I just want to leave you with some resources. So please go read the preprint if you haven't already. The code base is up on GitHub, so if you want to use the model and play around with it, it's there. And we also have this blog post. So yeah, so now I'm happy to take any questions or maybe.
All right, thank you so much, James. So let's answer some questions from the audience. Okay, so a colleague Pavan from Civics, right? He asked a question. ESM2 embedding is a great idea. We are seeing the same themes in other single cell formation models too, like State from Arc Institute. Do you pre-compute these across the cross species transcriptome before training, or so that you get backup genes to the ESM2 C, or or it's just a lookup, right?
Yeah, we do pre-compute them before training, and actually, if you download the model, you can download. I think there's over 20 different pre-computed ESM2 embeddings for 20 different species. And then recently, we're working on a update to the code base right now that has code for creating your own. So if you want a species that's outside of that 20 or so, you can now have we now have a script basically for generating that, and so you can pre-compute your own on any species you want.
Yeah. So one of my colleagues, Sin Lu from Amgen, actually have questions. Will the cells and the gene embedding be affected by the compositions of cell types? For example, some cell types are abundant and some are sparse. Will the cell and gene embedding are dominated by the abundance of cell types? And the also related to that, she asked another questions of how does TranscriptFormer perform in predicting similar cell states, such as specific fibroblast cell state beyond major cell type separations. Additionally, what occurs when a cell state that is not included in the training data encounter?
Yeah, two great questions. So the first one I think is about the gene embeddings and how much they're dominated or influenced by the cell type. Um, and yeah, quite a bit actually. But it actually does, as we saw, depend a little bit on the specific gene. So some genes tend to be more influenced or dominated by cell type than other genes. And I think that's actually a very interesting thing we haven't really probed very much yet. Um, but cell type tends to be one of the strongest biological signals we see in the model, and so it tends to dominate. Another thing we haven't tried is taking the gene embeddings from lower layers in the model, which presumably wouldn't actually be as dominated by cell type because there's less mixing that happens. But yeah, generally the context, which is cell type, tends to dominate at the later layers. And then so the second question was about cell state and generalizing to different cell states. And um, we do actually see that it does a very good job of generalizing. So we tried, the perturbation results and the disease perturbation was kind of around trying to understand how well cell state kind of generalizes. We haven't looked a lot at developmental um, cell states and changes, but I think we do see some evidence there from spermatogenesis that we do get quite a bit of separation from different stages of development. And I'll also say that we've seen that it generalizes quite well to new cell states. Like the Tahoe data set is actually all cell lines, right? And it wasn't trained on cell lines. It was trained on cell by gene data. So, we can get very different cell types in there or cell states in there, and it still seems to perform quite well. So it's great to see that it generalizes, and I think that's probably largely because it's trained on such a diversity of cell types across different species, and then the ESM2 embeddings I think also help there.
Right. Um, the next question related to that, probably let me, okay, this question is easy. So, how did you handle cross-species gene ontology mapping? Probably you're not doing this. Just right.
Yeah, no, we don't do any mapping like that. We simply kind of let ESM2. Um, so right, like when you embed two species with ESM2, there is already some kind of innate similarity. Like it is embedding it into the same space. These two species are now embedded into the same representation space, and so we don't have to explicitly do this mapping. We would kind of like you know the model to implicitly do the mapping or something, but yeah, we don't explicitly do this. This is we're more hoping this is to be some like a emergent property of the model.
Yeah, thanks so much. Another question from Bishop, colleagues from UIUC. Uh, could you talk more about the choice of GPT style casual masking over a BERT style of random masking?
Yeah, this is a good question. So yeah, a lot of other models actually use more BERT style masking,
You didn't
and
talk about that, particularly.
yeah, pretty much all of them. I think there's SE Mulan, which uses also the GPT style, but yeah, most of the other ones use BERT. And we really wanted to do this generative modeling aspect where we could sample repeatedly and generate data. And so you kind of need to do this in a sequence. And so that's one of the main reasons we decided to use the causal masking. I do think there are other good arguments to be made for causal masking, like that it trains more efficiently. Because when you train with causal masking, every single output gets a backpropagation signal, right? Because every single one has some value it needs to predict. Whereas when you use Brit style masking, this actually isn't the case, right? You completely mask out some of the positions, and so they don't get any updates in it. So generally, causal masking tends to be more efficient way to train the model. Um, but there are other techniques like a mass schedule. I think ESM3 started using mass schedules, which we haven't explored that, but I think that may be like another alternative that also allows for masking out most of the sequence.
Yeah. So the next question I think quite interesting that from Amanda. Have you tried using TranscriptFormer to predict cell state along a trajectory path? For example, predicting the gene expression of a cells that would be found between state X and state Y. You know?
We haven't. I do think also that that'd be interesting. Um, it's a little bit unclear to me that this would be exactly the right model. We haven't tried it, so we don't know how well it work. But VAEs tend to do things like that very well because they can interpolate between two different positions in a latent space. Um, but I think you need that probably need that Gaussian prior there to get good interpolation in the embedding space. It's usually not quite as good in a transformer, strictly transformer based model like this, but it could work. We just haven't got around to trying it. I think it's a really interesting direction.
Right. So there's actually a question in regard to engineering. How easy is it to use your tool? And another saying, okay, try it this, check it once and it worked and failed early this morning.
Oh no. Well, if it does fail, so first of all, I hope it's easy. But if it does fail and you have problems with it, please leave issues on the GitHub. Like we try to get to them pretty quickly, and we've already resolved over 40 of them. So like yeah, if you have any issues, please just leave an issue and we'll get to it. But it's supposed to be as easy as possible, or at least the CLI is supposed to be easy, and we've already made a lot of, I think, improvements in terms of making that um, easy because one of the main issues we have, we did a whole bunch of benchmarks, so we've seen many foundation models, and so we've run into many issues with different data types, and the problem is that every data set is different, and they're not all formatted the same, and when you input a new data set, often it causes problems. And so we've done some work on the CLI to make sure that it does all these kind of basic checks and so forth to format or get the data properly so you don't have to do the work. So if you're using TranscriptFormer right now, I encourage you to use the CLI, which is probably the best way to use it at the moment. We're also working on a Python client, which will probably make it easier to use like in a Jupyter notebook. But yeah, please leave
And
issues and we'll get to it.
right. And the last questions actually from a student. Can you elaborate your path moving from a one who's studying physics to the one who's used computer to decode?
Uh, yeah, sure. Yeah, it's not I guess a typical path. And so I studied physics for my PhD, and I studied particle physics, and I worked on one of the LHC collider experiments called Atlas. And Atlas collected just enormous amount of data coming off the accelerator, and it's actually a very big challenge is to analyze that data. So at the time we were looking for particles like the Higgs boson, as well as more exotic supersymmetry particles. And so I worked on the analysis part, and we used a lot of machine learning techniques to analyze the data. So at the time, this was like 2012-ish, and so deep learning was starting to become a thing. So, applying deep learning techniques as well as more traditional machine learning. Um, and so that's when I started to fall in love with the field of machine learning. And so after my PhD, I decided to come here to Silicon Valley, where I am now, and find a job. And I started working in data science, then more machine learning and AI, and eventually, eventually I was working at Amazon and mostly at Alexa AI. And that's when ChatGPT kind of exploded on the scene and became this really big deal. So we've been working on similar language models as well. But it really became apparent just like how powerful the technology was at that time. And so I really want to apply that technology to basic science research and questions. Wasn't so interested in building another product again. Not that there's anything wrong with that, but I still had this interest in science. And so I came to CZI because it's, what better field than biology to apply these new AI techniques because we have so much data and it's being collected so quickly and we have so many interesting problems to work on. So yeah, here I am now and I'm really enjoying it. And yeah, I
The
think
problem
this is just the
is so
start
much
of things to
important for
come.
us, right? The problem
Sorry?
is the problems is just so important for us, for each
Yes,
of
no,
us.
yeah, exactly. And the problems are really important, really impactful. I think we're still in the GPT-1, GPT-2 level of models right now when it comes to at least single cell data. And we haven't scaled it up to GPT-3 or 4, which is when people really started to care about the models and they really started doing really interesting things. So I think, in a few years, this field in this area is going to be really impactful.
All right. Thank you, James, so much for
All right.
that
Well,
great,
thank
great.
you. And thank you, everyone, for showing up and listening to my talk.
Thank you so much, everyone. Have
Okay.
a good.
Thank you.
Bye bye.
Bye.